Check out what I found. Ask and you shall receive. We're in the big time now, folks. All right, I've got hopefully a couple short videos for you tonight, and that will be the end of the material for the quiz that's actually going to be for not this week, but next week. Right now, just a short video kind of that's about one particular form of a statistical test that's used commonly in genetics, something called the chi-squared test chi-squared goodness test fit. Okay, so kind of calling back to some of the lectures that we covered last week, we talked about one of the sort of big red flags leading to the idea that genes may be inherited in a non-Mendelian fashion was some presence of unexpected results. And these can take different forms, right? They can take the form of actual new phenotypes, or they can actually take the form of, more commonly, sort of unexpected phenotypic ratios in the progeny. Okay, and that's really good what we're going to focus on here. Okay? So these unexpected ratios of phenotypes in our progeny serve as this red flag, and really then indicate that there's some possible non-Mendelian inheritance going on in the genes that we're studying. Okay? So we want to kind of dive into that in a little bit more depth and actually give that a little bit of mathematical rigor, quantitative rigor. Okay? And so one of the things that kind of drives this whole this test is really the question of whether or not these differences that we see, these unexpected results, whether that's actually due to some sort of non-Mendelian inheritance, or if this is really just due to chance. Remember that random chance plays a pretty large role in inheritance, depending on sort of which gametes fertilize and sort of which allele of a particular gene sorts into a particular gamete. Okay, so how do we differentiate an actual different pattern of inheritance from random chance? And so that's going to be what we talk about today. Okay, here it is. Whoa! Tell you what, that's just magical, folks. All right, so this brings us to our statistical test for the day. So it's the chi-squared goodness of fit test. Okay, so you'll often see chi-squared abbreviated. I think it looks like a funny X, something like that. I may oftentimes just use kind of a big X squared as a means of saying chi-squared. Okay, so it's the Greek symbol chi. I probably have that wrong. Okay, so the big question that drives this statistical test is, do our observed, I'm going to call this the O, results, do our observed results fit with our expected results? We'll call this the E. Okay, so it's a goodness of fit test. How well does this set of data fit with this set of data? Okay. So the basis of this test is then going to be the difference between the observed and the expected. Okay. So basically what this test does is it measures how well our observed conforms to what we expected. So importantly, this test takes into account sample size. Sample size is very important. Okay. I mean, it's largely true for a lot of statistical analyses, but it's very important for this one in particular. And so in this case, when we actually do our calculations, we're going to use the actual raw number of progeny in all of our calculations. Okay, so we're not going to use anything like proportions or percentages. Okay, so as you can imagine, it's important for this. So if you take, I think your textbook does a really good job of laying out the example. If you take sort of 10 coin flips, right, and you end up with 6 heads and 4 tails, that's not that far off kind of what you would expect. You expect 5 and 5. But say you do that 1,000 times, okay, and you end up with 600 heads and 400 tails. Okay, that actually is a much more significant difference, and also much more unlikely if there's an actual 50% chance of you getting a heads or a tails when you flip a coin. So if you're getting like 600 and 400, something's off with that coin. Okay, and that's really kind of what we're talking about here. So in this case, sample size is important, which is why it's advantageous for someone like Mendel to use a pea plant that can make lots of progeny. It's really advantageous for someone like Thomas Hunt Morgan and Alfred Sturtevant to use Drosophila because they can make thousands of progeny. They can actually get a really nice, not just measure of, say, the recombination frequency to estimate distance of genes on a chromosome, but they can actually go through with something like the chi-squared goodness of fit test and actually measure the statistical significance of whether or not these genes may or may not be linked. And that's going to be the example we run with at the end of this lecture. Okay, so a couple more details about this chi-squared test, goodness of fit. Chi-squared goodness of fit test. I will keep messing that one up. Okay. So basically, this test works. It never confirms a hypothesis. Instead, what this is done, what it's used for, it's used to reject a hypothesis. Okay. It's going to test a particular hypothesis. And so the hypothesis that we use in this in the case of a chi-squared test is the null hypothesis, N-U-L-L. In essence, the null hypothesis going into any chi-squared test is that the observed data fit with the expected data. Okay. So in other words, if we're studying if our idea is that two genes are inherited in a Mendelian fashion, and that's what we base our expectations on, the null hypothesis is that the genes are inherited in a Mendelian fashion. Okay? So this is our null hypothesis. We kind of make this before we go into the chi-square test, and we do the chi-square test, and we decide whether or not we're going to accept or reject this null hypothesis and then move on. Okay? So if we can reject this null hypothesis, that our observed data fit with the expected, that our two genes are inherited in a Mendelian fashion. If we reject that, that gives us a little bit more of a quantitative basis to then kind of form a new hypothesis that maybe these genes are linked. Okay? So we'll kind of revisit this at the end, but this chi-square test never actually proves that two genes are linked. It just gives us a little bit more quantitative basis to sort of suggest the likelihood that they are linked. Okay, so the final result out of any chi-square test something called a p-value. Okay? So this p-value is a probability. Okay, but it's really the probability that the difference between our observed and expected due to chance, okay? Chance, not change. Chance, okay? I'll kill this real quick down here. Okay, so if what we get out of this is a high p-value, it means that there's a high chance high probability that the difference is due to chance. And this leads us to accept the null hypothesis. Okay? Now if there's a low probability, or a low p-value, rather, this indicates that there's a low probability that the difference is due to chance, okay? And then this allows us to then reject our null hypothesis. Okay, rejecting this idea that there's no difference between the observed and the expected. Okay, so how do we get to this p-value? That's what I'm gonna want to walk through right now. Okay. So your textbook, I think, it goes through. I think it's a really good illustration of one of the main reasons why this chi-square test is used. I've already mentioned this. It's often used to test whether or not two genes are linked, or to investigate whether two genes are linked. The example they go through, they don't explicitly mention it, but I think that the cross that your textbook does in the example is actually homozygous dominant in two alleles crossed with homozygous recessive in two alleles. Okay, so that leads to, they've got their parental genotypes in the gametes of homozygous dominant, homozygous recessive, and then they've got the recombinants listed like this. So just for clarification, if you want to walk through the example the textbook gives you, I think this is the cross that they used to do their example. Okay, so I'm going to use a different example to walk through our example chi-square test. And since we've really already kind of talked a bit about these hypothetical genes and their hypothetical linkage, we'll go back to the slugs for this one. And we'll look at the coat pattern gene. Okay, this is the one that was regulated by the big S or little s gene. This is the one where this is the dominant S allele coded for a striped coat. This recessive S allele coded for a solid coat on these slugs. And then we thought that there may have been some linkage between this coat pattern and antenna shape. Okay, the two alleles for this gene were the dominant C, the big C for curved antenna, and the recessive lowercase c for straight antenna. Okay. So we thought that these coat patterns, that the gene for coat pattern and the gene for antenna shape were linked. So this leads to our null hypothesis. Okay. That the genes for coat pattern and sorry, not seeds, antenna shape are not linked. Okay, that's going to be our null hypothesis going into all this. Okay, and so we did an example. We did a dihybrid cross, and so a dihybrid cross in this case was two hets at both alleles. Okay, the other way we wrote this after kind of knowing that they were linked was like this. I already messed that one up. Up top, our standard notation for this one is SSCC. And here's our sort of new linkage annotation to sort of indicate that these two alleles are the same chromosome. I apologize for the confusion on that one. Okay, we did this cross and we had a total of 160 progeny. Okay. So we had four expected sort of phenotypes out of this one, phenotype classes. Okay. Both dominant traits, one dominant trait, other dominant trait, both recessive traits, or both recessive phenotypes rather, so both dominant phenotypes, one dominant phenotype, the other dominant phenotype, both recessive phenotypes, and we expected to see these in a 9 to 3 to 3 to 1 ratio. Okay, so with a total of 160 progeny, our expected for this one was 90, 30, 30, and 10. I made the math pretty easy. Okay, and let's say we observe something different in this case. So we're not going to use the same data set we did from last week. So let's say in this case we observed 101 that had both the dominant phenotypes. We observed 19 that had a striped coat but straight antenna. We observed 23 that were solid but had curved antenna, so that's the other dominant trait. And we observed 17 that showed both recessive phenotypes. Okay, so they were solid with straight antenna. Okay, so I changed the data up here just to illustrate kind of why this test is important. So if you look at these data right now, they're not quite as obvious last time that we're off of the expected phenotypic ratios. Okay, so 90 is certainly different than 101. 19 and 30 are certainly different, but they're not as obviously different as I made them out to be last time. Okay, so I did that on purpose to sort of show you why someone would use this, because it's a little bit harder to say right here that, oh yeah, definitely these don't sort of these don't show the same ratios that we expected. Okay, you don't have you don't have as solid a basis to say that. So you actually need to go through here and give others a quantitative measure through which you can state that we think that these don't conform to our expected ratios. Okay, so that's kind of our cross leading into this. So let's actually show, I'll actually put down now the equation. So the chi-squared equation. Okay. So in this case, chi-squared equals, this is Greek symbol for sigma, which is a way of saying the sum of, okay? And so this is we're gonna add up a bunch of numbers in this case, and what we're gonna add up are a bunch of number observed minus the number expected squared over the number of expected. And we're going to do this for each phenotype class. And we got 4. 1, 2, 3, and 4. So in this case, we're going to calculate 4 of these. We're going to add them up, and that's going to give us a number for the chi-squared test. Okay, so I'm going to go ahead and add some more columns here. Okay, so we're going to add an expected minus observed. That's this, or I guess we can do O minus E. It doesn't really matter. Let's do it the right way anyway. Observed minus expected. Okay. The next column we're going to put in there is O minus E squared. Okay. Then the last one we'll do is O minus E squared divided by the expected, okay, which is actually what we want to add up for each of these phenotype classes. So we're going to go ahead and do these for kind of all 4 of these. So we just kind of quickly go down through. This first one is observed minus the expected. So this is the actual difference. Okay, this is the basis of how this chi-squared test works. It's going to actually look at the difference between the observed and the expected. So this is 11 here, 11 here, 7 here, and 7 there. Okay, there's our difference. Okay, now we're going to square that difference to make those differences pretty large, right? So 11 squared is 121. 11 squared is the same, 121. 7 squared is 49, and 7 squared is 49 down right there. Okay. And the last calculation then is to take this number here and divide it by the expected. Okay, I've already done the math on this one. And so if we take 121 and divide it by 90, we get 1.34. 121 divided by 30, 4.03. 49 divided by 30 is 1.63. And then 49 divided by 10 is 4.90. Okay? So these are 1, 2, 3, 4. These 4 numbers, or this number, for each of these 4 phenotype classes. Right? So now we just want to add all these up to get our chi-squared value, because that's what this sigma here means, the sum of all these numbers. Okay? So if we add all these together, we actually get 11.90. Okay? So this is our chi-squared number for this data set, 11.90. But I remember I told you the final product for this chi-squared test is a p-value. So how do we get from this chi-squared value to the p-value to get our final measure of probability? Okay? So I'm going to hold on to that number, but I'm going to get rid of all this, and we'll talk about the last step in the chi-squared test. Okay, so our chi-squared 11.90. Okay, what the heck do we do with that one? Okay, so what I want you to do is take a look at table 5.2 in your textbook. That's actually page 155. I'm going to draw out like a simplified version of that just to show you, but I want you to follow along with this in your textbook. Okay, so this works. The very left-hand column of this table is something called the degrees of freedom, or DF. Degrees of freedom. So really simply, this is the number of classes minus 1. Okay. So in our case, we've got 4 different phenotype classes. So we're going to take 4 minus 1. So for us, it's 4 minus 1 equals 3. Okay. Because we've got 4 different phenotype classes we're working with. Okay. So the very left-hand column of this is degrees of freedom, and it goes from, I think, 1, 2, 3, 4, 5, but we're going to focus in on this row here with 3. Okay, now on this top row here, they list out the p-values. Okay, so this is where we're actually going to get our p-value out of this thing. Okay, so we got some example. They got p-values. They don't kind of list a whole continuum, but they got some different different thresholds here. So 0.99, 0.90. They put down 0.50, 0.10. Then the color changes. We get to 0.05, 0.01, and 0.001. Okay, so again, let's think back to what this p-value is. It's a probability that the differences that we see are due to chance. Okay, so the higher the p-value means more likely due to chance. Okay, lower p-values more likely due to inheritance. Okay, now generally within this statistical test and actually a bunch of others across all the sciences, the main threshold for statistical significance is a p-value of 0.05. Okay, so we often draw kind of a big dividing line right there. So the cutoff is usually 5%. Okay, so if you can show someone in any data set that there's a 5% chance, or there's less than a 5% chance that the differences you see are due to chance, people typically believe that. Now, smaller p-values are better. That means there's an even smaller chance that difference is due to chance. Okay? Smaller probability that difference is due to chance. Let me clarify that. Okay, so then let's look at the table, and I'll just put down some numbers on this row 3, because that's the degrees of freedom we're working with. So we've got 0.11, 0.58, 2.37. Notice how these are getting bigger. So the bigger the chi-square value, the more likely it is that the difference is due to inheritance and not chance. 2.37, 6.25. We get past our threshold, 7.81, 11.35, and then the last one in this row is 16.27. Okay. All right. So this is our chi-squared value, 11.90. So as we look into this one, we kind of go through here. Basically, we want to find the point at which our chi-squared value is bigger than the number in this row. So we keep going along, and all of a sudden we get to here. So we know that our chi-squared value of 11.9 is greater than 11.35. It's also less than 16.27, so we can't go past that threshold here. But what this tells us, because our number is greater than 11.35, this means that the p is less than 0.01 in our case. Okay, so there's less than a 1% chance. There's a less than a 1% probability that the difference between our observed and our expected is due to chance. Okay, so there's a high likelihood that this is actually due to some sort of funky inheritance going on. Okay, so we can't prove that the inheritance is due is because these two genes are linked, but we're able to reject our null hypothesis. Okay, so we can reject the idea that these two genes are inherited in a Mendelian fashion. Okay, and then we can kind of move on from that. Okay, so this is essentially how a chi-square test works. So again, I want to remind you that this doesn't in any way prove that the two genes are linked. You have to kind of continue and do more experimentation to figure that out. But it serves as that first quantitative basis through which you can reject the first idea, which is that these genes are inherited in a Mendelian fashion. And now it's often used to test gene linkage, but it can also be tested for other things. We didn't go through any real examples of this, but if you had a particular trait and you wanted to differentiate whether or not this trait was regulated by one gene or maybe two genes, that could give you some different expected phenotypic ratios, and you could show statistically that it's not possible that your gene that your trait is regulated by one gene, and instead it may be regulated by two genes. So you can kind of continue on down the line that way. All right, so that's the chi-square test, chi-squared goodness of test fit. Know it. Thank you.